Hello, everyone, and welcome to today's live broadcast, Good Culture for Your Cells, Best Practices for Preventing Contamination, presented by Dr. Jessica Wagner, Application Specialist, Cell Handling, Eppendorf AG. I am Alexis Cross of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Eppendorf. For more information on our sponsor, please visit eppendorf.com. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Answer a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our presenter, Dr. Jessica Wagner. Dr. Jessica Wagner is an application specialist focusing on cell handling at Eppendorf headquarters in Hamburg, Germany. Entering a cell culture laboratory as a graduate student for the first time in 2006, she came to Eppendorf with 5+ years of experience in cell culture. For a complete biography on Dr. Wagner, please visit the Biography tab at the top of your screen. Dr. Wagner, you may now begin your presentation.
Thank you, Alexis. Welcome also from my side, and thank you all for joining us today. The presentation will take about 45 minutes, and we will take care of your questions right after it. So let's start now with our webinar by having a look at the contents. First, we will briefly touch on the types of contaminants, because if you fight something in the lab, you would want to know what you're fighting against. We will also speak about contamination sources and some easy ways to prevent contamination. At the end, we will go through just a couple of tips for aseptic handling. Contamination is an everyday challenge in a cell culture lab. All labs are prone to contamination, but the frequency varies. All labs have contaminations from time to time, but understandable, nobody likes to talk about it. I think we can also agree that contamination costs time and money, and undetected or even ignored contamination actually endangers the reliability and reproducibility of our results. So what do we do against it? In the majority of cell culture labs, 70% ethanol is the #1 substance for disinfecting surfaces, equipment, etc. The good thing about ethanol is that it is very effective against bacteria, yeast, and some viruses, and it evaporates completely residue-free. Therefore, it is well suited for routine rapid disinfection, for example, of the work surface in the biosafety cabinet and equipment that is used in the hood. If possible, you should prefer wipe disinfection over spray disinfection, not only to prevent aerosols, which are harmful when you inhale them, but also to enable a complete wetting of the surface for a proper disinfection effect. One of the drawbacks of using ethanol is that it is not effective against all kinds of contaminants. Bacterial spores, for example, can survive alcoholic solutions. And although ethanol has a lethal effect on fungi, it takes a few minutes until the fungi are killed. And due to the fact that ethanol evaporates within seconds, it is not really effective against fungi. So if you are facing ongoing problems with fungal contaminations, it may become necessary to implement a cleaning routine with a disinfectant which has a broader spectrum. The second thing many researchers worldwide use to fight contaminations in cell cultures are antibiotics. This used to be a good tool, but it is becoming more and more a poor tool because there is limited protection due to the increasing resistance of bacteria. The most commonly used antibiotics in cell culture are penicillin and streptomycin. Their non-cytotoxic effect was first described in the 1940s, and they are still present in a lot of cell culture labs worldwide. As you can imagine, in over 70 years, the continuous usage of these substances supported the increase of resistant bacteria tremendously. So does it help to simply switch to other antibiotics that are not so common? Well, it may be a short-term solution in some cases, but in the end you are facing the same issues with resistant bacteria. A more general aspect of the routine use of any antibiotics in cell culture is that it gets more difficult to detect microbial contamination. Without antibiotics, most of the introduced contaminants would soon be visible under the microscope or even with the naked eye as color changes and turbidity of the culture medium occur. The prophylactic routine use of antibiotic in cell culture not only masks poor aseptic techniques, it can also leave low levels of partially resistant contaminants undetected for longer time periods, and therefore creating a greater risk for the whole lab. Besides the effect of antibiotics on bacteria, what most people ignore is that it is known that antibiotics can have side effects on the metabolism of cells. And these two studies here on the right are just examples. So here we have a list of various papers describing the effects of antibiotics on a number of very different cell types. So taken together, antibiotics are not really good in protecting the cells from bacterial contaminations, and they are not really good for your cells. And this is why we would like to show with this webinar that there are simple and efficient ways that offer huge potential to protect your cell culture from contamination. So before we start with the next chapter, we would like to ask to participate in our first poll. So the question is, did you ever have problems with contamination in your cell culture lab? And the answers you can click on are never, sometimes, or on a regular basis. So please click now to vote. Okay, and the answers are coming in. So I'm going to send the results now to you. So as you see, the majority of you, more than 80%, has to deal with contamination sometimes. Even 10% do see contamination on a regular basis. So it is still a common problem in cell culture labs. So let's start with the second topic. Let's see who our enemy is. In general, we find two major types of contamination, the microbial ones and the eukaryotic contaminations. The microbial ones are fungi and yeast and bacteria, and here a special type of bacteria, Mycoplasma. The other ones, the eukaryotic contaminants, are not necessarily so well known and are cross-contamination of other eukaryotic cells in cell lines. Now we want to look at all of these in more detail. Let's start with the more common types of contamination, fungi, yeast, and bacteria. In most labs, it is one of these three types if there is a contamination. Bacterial contaminations, as seen here on the upper left image, are in most cases easy to detect as they usually overgrow cultures very quickly. In infected cultures, you typically see an increase in turbidity of the medium and a color change to yellow, as seen here on the lower left image. Sometimes there is a thin film on the medium surface visible as well. Under the microscope, the bacteria appear as tiny, in most cases moving spots between the cells. Fungi and mold can appear as small isolated colonies of gray, white, or greenish color floating at the surface of the medium, which you see here on the lower left, on the lower right image. It is important to inspect the culture vessels with the naked eye and look for colonies which rest on the medium surface and will be missed during microscopic inspection, especially when you work with, for instance, big tea flasks. This is something that can be easily missed then. You can see such colonies here, as I said, on the lower right image. And if the contamination is substantial, the medium will become turbid and cloudy, and spots on the vessel surface may appear. Sometimes fungal contaminations will cause a pH increase of the medium, resulting in phenol red containing media to appear pink. Yeast contamination, as you see here on the upper right image, are unicellular fungi, and under the microscope they appear as round or ovoid bright particles. They can also exist in chains of 2 or more particles and are sometimes even multi-branched. Initial stages of yeast contamination are difficult to detect with the naked eye because the pH changes only slightly and initiating little or no color change in the medium. The next and not so nice contaminations are a special type of bacteria called mycoplasma. They are very small and have no cell wall and grow as parasites attached to the cell membrane. The bad thing about these guys is that they are not detectable in light microscopes. So even if you monitor your cell lines regularly, you wouldn't detect them. They don't float in the medium like other bacteria, but are attached to the cell walls, to the cells. That makes them somewhat invisible. In order to find out whether your culture is clean or contaminated, you need to check the culture, for example, via PCR. Mycoplasma secrete toxins that are not good for the cells. And the other point is they grow very fast, actually faster than the cells, and therefore competing for nutrients in the medium. So your cells might actually starve due to mycoplasma contamination. And this is another reason why a latent contamination goes undetected. If you are on a good feeding schedule, your cells actually might not suffer, although there is a persistent mycoplasma contamination. I want to go a little bit deeper into the topic of how mycoplasma affects cellular metabolism by showing you some data from a study published in 2013. In this study, HeLa cells were infected with mycoplasma, followed by analysis of differential gene regulation up on different time points of infection. And as you can see in this graph, the number of genes that respond to the mycoplasma infection increases dramatically with time. And I just want to briefly show you an extract of the list of genes that were identified. I know it's not so good to read, but let me tell you that the cellular effects were dramatic. So the genes that were deregulated upon infection are associated with signal transduction pathways, immune responses, cell cycle regulation, cell growth, and other cellular functions. So I think that we all agree that mycoplasma contamination can have dramatic effects on cell cultures. And now I want to switch to another type of contamination. Cross-contamination means that a given cell line is contaminated by another cell line. Usually this happens by an accident somewhere during the handling of cells, so it is a mistake in aseptic handling. For example, you are supposed to trypsinize one cell line after the other, but since you are a little bit in a hurry and you handle another faster growing cell line depicted here in pink at the same time under the hood, by accident a little droplet of the more rapidly growing cell line gets into the culture shown here in green. And because the pink cells grow faster than the green ones, with the following steps of cell culture, the pink cells become more numerous and start to overgrow the green culture. After a given number of subcultivations, you will end up with a dish of pink cells or a mixture of both. So, for example, if the green cells would be Vero cells and the pink ones would be HeLa cells, you would end up with a dish that is labeled Vero but actually contains HeLa or both of them. As soon as two cell types are mixed up, it is very hard to distinguish those two. This process goes fast and it might go undetected, especially if the morphology of the cells is not drastically different. We know this problem already since 1967, when the first publication came out. Still to date, we know that 15 to 20% of all cell lines used worldwide are either cross-contaminated or misidentified. Currently, there are more than 400 cell lines known to be misidentified or cross-contaminated. So what all of those types of contaminations have in common are the consequences: loss of time since you have to redo experiments, increase in cost, time is money and more material that you need, loss of reproducibility. You might have completely different responses when you work with contaminated or non-contaminated cells without knowing it. And the last point here might be the most horrific: the loss of significance. So if you want to find a new therapeutic treatment for breast cancer, but you work with melanoma cells without knowing it, your work is useless. So in order to efficiently fight the contamination, we need to know where it comes from, and this is what we look at in the next chapter, sources of contamination. I want to distinguish between the indirect and the direct contaminations. Indirect contaminations come from the labs or the equipment or even from ourselves. The direct contaminations come from reagents or you use cells that have been contaminated before you took them in culture. So we start with labs and equipment. You might have airborne germs that come from ventilation and opening the doors, hopefully not from opening the windows, which should be strictly prohibited in a cell culture lab. Then we have the water that might bring in the germs. This can be water from the tap, the water bath, or the water tray inside the incubator. The water might carry two different types of germs. First, the primary germs that are brought with the water in the lab, for instance, in the tap water. But also you have secondary germs that were originally airborne. For example, in the incubator tray or the water bath, you would probably use distilled water or autoclave it before use, so it is free of waterborne germs. But the water is in contact with the air, and germs might literally fall into the water with the air. So the next source for contamination is dust and dirt, which is a nutritional base for germs. So the more dust you have, the more ground you have for potential contamination. The next indirect source, maybe the most important one, is the personnel. We all carry bacteria. They belong to our flora, on our skin, in mouth and nose, and they can affect the cell culture. When we are ill, we even carry more germs with us. The clothes we wear underneath the lab coat are the clothes we are wearing on our way to work, and they have been in contact with the outside air and the other sources of contamination. So some microbes might stick to the clothes and we carry them into the lab. Same is true for the hands and other skin parts. Apples, for example, can have large amounts of yeast on the surface, which is not a bad thing if you eat them. But if you don't clean your hands before entering the cell culture lab and start to work, this might affect the cell cultures. The shoes are also a major source of contamination. They can carry traces of soil and dirt, and we carry the microbes that are in there in our cell culture lab if wearing street shoes there. So let's move to the direct sources, the reagents. We use a broad range of reagents in cell culture. Media usually are rarely delivered contaminated nowadays. They are sterile filtered, and it happens very randomly that you receive them contaminated. FBS used to be a very important source for contamination, especially for mycoplasma. The situation has greatly changed in the past, since most production facilities usually filtrate FBS now at 0.1 microns, which filters mycoplasma more efficiently, just not as efficient as other bacteria due to their small size. Also, mycoplasma don't have a cell wall and are therefore quite flexible. In addition, there is also gamma-irradiated FBS out there, which further increases the safety of the products. Production facilities and labs working under GMP, GLP conditions usually do routine testing on the FBS that is used. Last but not least, any aliquot prepared in the lab carries a potential risk of contamination. The person preparing the aliquots might have made a mistake in aseptic handling, and your stocks might carry a contamination. So before I go to the next slide, I have another poll question for you and would like to ask you to participate by clicking. So the next question is, what kind of cell types do you use in your lab? And you can click on cell lines, primary cells, or both. Okay, and again, I wait a few seconds until the answers are coming in. And I'm going to now send the results to you. Okay, so we see almost 40% of you are working with continuous cell lines. Almost 15% is using only primary cells, and the majority of you, almost 50%, is using both continuous cell lines as well as primary cells. So let's first talk about the risks when working with cell lines. If you introduce a new cell line to the lab, you have a potential risk of introducing a contamination. So you also might rely on older stocks that simply never have been tested. And in addition to microbial contamination, misidentified or cross-contaminated cell lines are a threat. As mentioned before, 15 to 20% of all cell lines used worldwide are estimated to be cross-contaminated or misidentified. For primary cells, the situation is a little bit different because they are finite. They die off at some time point, and you have to generate or purchase new cells. Primary cells often are contaminated due to the semi-sterile tissue preparation. There is no possibility to sterilize tissue samples, and in most cases you just place them in a sterile container. Upon cultivation, you have to make sure that you don't get microbes into your freshly prepared tissue, so your primary cell cultures always carry the risk of showing microbial contamination. With primary cells, you have hardly the risk of cross-contamination simply because they have a limited lifespan. In case your primary cells suddenly become immortal, then you should think about cross-contamination. Spontaneous immortalization is hardly seen in cell culture. Of course, you should validate that you isolated the cells of interest. Again, cell authentication is an important factor and should be considered as a routine step in cell culture. So we know now who our enemy is and where it comes from. So the next logical step is preventive hygiene, but before we start, here comes our next poll. I'm interested in how you learn cell culture techniques. Okay, and the question is, how did you learn cell culture techniques? The answers you can click on are learning by doing. My colleague in the lab taught me how to work with cells, or I attended a practical training course or a lecture. Okay, and I wait a couple of seconds and then I send the results to you. Okay, so we see that the vast majority of you, almost 80%, learned their cell or their aseptic techniques from a colleague in the lab. So this is not really surprising. It confirms that standardized training courses or lectures for cell culture techniques are not so often seen, and the majority of us is still learning from our colleagues, which can be a good thing, but unfortunately can also be a way of passing on bad habits or obsolete techniques, especially when not all members of one lab follow the same standard operating procedure. Okay, so the next chapter is then preventive hygiene. From this slide, you may recognize that it looks similar to the first slide of the previous chapter. We have indirect measures of preventive hygiene and direct measures. We divided the chapter in this way because we would like to convince you that you should not take precaution individually for each type of potential contamination source. There is not the one way to wipe out the risk of contamination. So for every source, you should choose a special approach for preventive hygiene. Before we look at what we do, I would like to talk about a general principle, how to prevent any unnecessary transfer of germs into the lab. That sounds very easy, but a lot of labs don't actually do it that way. So here it is. If you work in a cell culture lab, you work under a biosafety cabinet, and within the hood there is a sterile atmosphere if you handle it properly. It allows you to perform your work in a sterile way unless you carry germs into it. There is a potential risk that germs get transferred into that sterile area, so a reduced germ load in the cell culture lab itself reduces the risk of carrying germs into the laminar flow. And so at the door to the cell culture lab, there should be some precautions to reduce the germ load. So this is what some institutes don't do. They treat their cell culture lab a lot of times like a regular lab, so the green area is also blue in these facilities. So before you enter the lab, wash your hands and disinfect them and use hand cream. Healthy skin has fewer germs than dry skin. Enter the lab and put on gloves immediately and disinfect them. Don't walk into the lab with street shoes. You can put a rack with lab shoes next to the lab door so people can change their shoes right before entering and right after they are leaving in the lab. There are also sticky mats available that you can put on the floor in the entrance area, and these will remove dust and dirt from your street shoes as well. Inside the cell culture lab, a separate air filtration system and a positive air pressure can help to keep airborne germs out when the lab door is opened. To limit the sources of contamination, only people who actually have to work in the lab should be allowed to enter. And personal protective equipment is a must. It is not only to protect yourself, but more to protect the cells. The lab coat, gloves, and shoes you're wearing inside the lab should be restricted to the cell culture and, if possible, use a gowning room or area close to the lab as a sluice where you put on your cell culture equipment. It's common that the complete lab is cleaned once a month, but of course this is depending on the lab, how big it is, how many people are working there. In the next few slides, I want to talk about the typical equipment that is found in every cell culture lab. So let's start with the CO2 incubator. Due to the door openings, the inside of the incubator is never sterile. Every time the door is opened, microbes can enter the incubator, and in addition, the atmosphere inside is disturbed. Therefore, door openings should be reduced in duration and frequency as much as possible to increase the contamination risk and to keep the atmosphere stable. A standard cleaning routine should be part of every cell culture lab. The humid atmosphere and warm temperature inside the incubator are not only necessary for the growth of your cells, but also support the growth of contaminants. Therefore, you should take special care about cleaning and disinfecting the incubator on a regular basis. If you have an incubator with a seamless, welt-free lining, you can simply clean the inside by wiping it with a proper disinfectant. If the incubator chamber has corners, fissures, seams, you should pay special attention to these areas and clean them, for example, with a clean brush. If you have an incubator with an automatic disinfection routine, you should run this at least once a month, or every 6 to 8 weeks would be recommended. And an incubator with copper interior also reduces the risk of contamination, as the copper lining reduces the bacterial growth. The water tray inside the incubator is necessary for keeping the atmosphere humid. The water inside the tray should be autoclaved and exchanged regularly because it is a source for contamination, as airborne germs can grow in the water. So instead of just adding water when needed, the tray should be emptied completely, cleaned with alcohol, and refilled with fresh, sterile, and distilled water. Adding any biocides to the water should be considered very carefully because they might cause corrosion and produce volatile compounds that might affect the cultured cells. If the incubator has a fan inside, the germs get actively distributed by the forced airflow, so an incubator with a fanless design will reduce the spread of contaminants throughout the complete chamber. Some incubators come with a UV lamp inside, which is supposed to decontaminate the circulated air. But before you consider paying for such a feature, please keep in mind that the high humidity inside the incubator adversely impacts the effectiveness of UV. So it might be not worth to spend the money, especially as it means recurring costs because the UV bulb has limited runtime. Let's come to another standard device in the cell culture lab, the water bath. It is used for warming media and reagents and for quick thawing of frozen cell stocks. Due to the temperature and the humidity, the water bath is an important source of contamination, so it should be cleaned thoroughly on a regular basis. Besides that, the addition of bactericides and fungicides to the water is recommended in this case. When you remove the vessels from the water bath, make sure that you disinfect them properly before placing them in the laminar flow, and pay special attention to the cap area when disinfecting because this is a place where germs can hide. A good alternative for heating aliquots of media, serum, or other liquids is a thermoblock that works without water and is therefore basically contamination-free. Let's talk about pipettes. All pipettes or pipetting aids should be restricted to the cell culture lab. A good tip is to simply put a sticker on each pipette and label it with cell culture so that everyone knows that this pipette should be kept inside the cell culture lab. Disinfect the pipette each time before you place them in the hood. In addition, you can disassemble the pipettes to clean it from the inside. For keeping precision, pipettes should be calibrated at least once a year. In general, all devices and surfaces in a cell culture lab should be wiped and dusted regularly. Wherever possible, use dust coats, for example, for microscopes. Spills should be cleaned immediately, and if available, use centrifuge equipment that is autoclavable. Despite the right handling and taking care of equipment, there are some do's and don'ts that you should keep in mind when entering a cell culture lab. Always wash your hands before entering and when leaving the lab. As mentioned before, you should have personal protective equipment that is restricted to the cell culture lab. It will help to keep germs under control that are on your clothes, shoes, or on your skin. Wearing a mask might sound a bit paranoid to you, but it can be a good idea in case you have, for example, a cold or another kind of infection. So let's move on to the direct sources of contaminations and the measures that can help to avoid them. Risky materials like FBS should be subjected to an intake quality control like you see here in this scheme. Only when you are sure that the new batch of FBS that you purchased is free of contaminants and your cells grow the same with a new batch, you can start working with it in your regular cell culture. You should retain a reasonable amount of the old batch for some period of time. This will allow head-to-head testing if problems arise when the new batch is introduced. As mentioned before, mycoplasma are a special threat in cell culture labs because they cannot be seen with a light microscope and they don't cause any visible changes of the culture media. In addition to the intake quality control I showed you on the previous slide, a test for mycoplasma can reduce the risk of a direct contamination through reagents. Choosing the right test method for your lab can be critical. The access to the desired equipment, the number of samples, and the time for getting the results are a major decision point. All detection methods have their advantages and disadvantages. You can download an overview from our website, eppendorf.com/cellexperts, if you want to find the ideal method for your lab. Media and reagents, especially serum, should be purchased from reputable sources which provide a certificate of analysis. Every aliquot of FBS, medium, or other reagents should be labeled with name, date, and lot number in order to backtrack if there arises any problem. Another direct source of contamination are the cells themselves. When you purchase new cell lines or primary cells, make sure that you get them from a reliable source like the American Type Culture Collection or the European Collection of Cell Cultures. When cells are given from lab to lab or when you isolate primary cells from tissue, these cells should be considered as potentially contaminated and be separated from the other cells until proven absence of contaminants. Reserve one shelf in the incubator for new cells, or if possible, use a separate incubator for quarantine. In general, it is a good advice to always keep primary cells and cell lines separate. We already talked about cross-contamination of cell lines and the importance of cell authentication. So before the next slide, I would like to start the next poll question. Have you ever encountered cross-contamination with a different cell line? So the answers you can click on are yes, no, or I don't know. And again, I will wait a few seconds until the answers are coming in. Okay, and I'm going to show now the results to you. Okay, so we see that 22% has seen cross-contamination with a different cell line in their cell lines. Almost 50% did not see a cross-contamination, and 30% is not sure about it. So please ask yourself if you've never encountered a cross-contamination in your cell line, if you actually do regular testing of the identity of the cell line. So we will go on. Cell line identity testing using short tandem repeat or STR profiling ensures you that the cells you're working with are the ones that you think they are. To be completely sure that the cell profile has not changed over the course of a study as a result of cross-contamination, you might consider STR analysis at the start, middle, and at the end of a project. So you should consider regular checking of STR profiles, especially for long-term studies with one cell line. Okay, so let's come to our next chapter. Here I want to give you some simple tips for aseptic handling. As for all other equipment in the cell culture lab, The hood should be cleaned on a regular basis, as this is the area where you open the dishes, plates, or flasks. There are some things to be kept in mind to avoid contamination. Disinfect the working area in the hood completely and let it dry before every use. Disinfect every piece of equipment that you bring in. Every item that is in the hood disturbs the laminar airflow that keeps the germs outside, so you should not overcrowd your working space with items that you don't need for that particular step. And when working at the hood, it is a good advice to have one clean side with culture media, pipettes, and new consumables, and the other side is reserved for liquid waste, used tips, and so on, like you can see here in the lower picture on the right. Your hands are the major source of contamination when you handle the cells in the hood. So disinfect the gloves sufficiently. That means let them dry before you start working. Give the disinfectant time to do its job. And disinfect them not only once but repeatedly. For example, when you adjust your glasses, never do it without subsequent hand disinfection. When you are pipetting, try to avoid moving your hands or arms over open vessels. In general, you should avoid hectic movements with your arms inside the hood, and when standing or sitting in front of the hood, as it will disturb the laminar airflow as well. To minimize the risk of cross-contamination, only work with one cell line at a time and prepare separate medium bottles or aliquots for each cell type. Before you start working with the next cell type, disinfect your working space in the hood. Just a few words about pipetting techniques. When you need a precise volume, hold the pipette vertical when aspirating the liquid. Pipette slowly to avoid the risk of contaminating the pipette itself. And when you're releasing the liquid, you can tilt your hand to avoid that your hand is directly over an open vessel. Pipette with a medium speed to avoid any droplets. Using filter tips protects the cells from being contaminated, and it protects the pipettes from being contaminated from the inside. So you might consider using filter tips at least for the very important steps like freezing of cells to build up stock cultures. In the last two slides of my presentation, I want to talk about the vessels that are in constant and direct contact with your cells, the cell culture consumables. When you open a plate, dish, or flask, the lid should be placed outside the working space, but in the hood, of course. Some people hold the lids in hand while pipetting, but this requires practice and routine because you have to avoid touching the inside of the lid while holding it, which can be quite tricky, especially with flasks. When you are placing the lid aside, you can position it inside up or inside down. Both ways are okay when performed correctly. That means when you put it inside down, make sure that the surface is clean, which you should do anyway. And when you put it inside up, make sure that you don't move over it with your hands or arms. When you use Eppendorf consumables, I would recommend to put the flask cap onto its side because thus it is not in contact with the work surface nor the airflow. Its anti-rolling design will make it stay in position. In addition, the Eppendorf plate lids have little support hubs that minimize the surface contact when the lid is placed inside down. The routine steps in your daily cell culture work include a lot of transporting the cells from A to B and back again. I give you an example. You take the cells out of the incubator, put them under the microscope to check if they are ready for splitting. Then you carry them to the biosafety cabinet, exchange the medium or split the cells, and then you bring them back to the incubator. And sometimes, for example, when you trypsinize the cells, you go again from the incubator to the microscope, to the hood, and back to the incubator again. So transporting flasks, dishes, or plates with medium inside always carries the risk of spilling. I already mentioned that spills, for example, inside the incubator should be cleaned immediately to reduce the risk of contamination. When using flasks, make sure that you carry them slightly tilted upwards to avoid medium contact, and in addition, flasks with a hydrophobic filter give you extra safety. Dishes with a corrugated gripping ring and plates with a gripping area give you the safety when transporting them, and in addition, the Eppendorf dishes have a special ring inside their lid that traps spills inside the dish. When you want to take one or a few dishes from a stack in the incubator, the gripping ring avoids unintended opening of the lid, and so does the gripping area of the plates. Before we come to the last slide, here comes the last poll question. So please let us know how you rate the content of this webinar. Okay, and so this is the end of the presentation. For more information on cell culture contamination, please visit eppendorf.com/cellexperts. Here you can also find information about webinars and on-site training courses for different topics, including cell culture. And yeah, so this is it for now. Thank you for your attention so far, and I will head over to Alexis again.
Thank you, Dr. Wagner, for your presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Answer a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, do you recommend using antibiotic AA-free media in order to detect contamination on early stages?
Yes, I would do so, because if you have, for instance, contamination of partially resistant bacteria, these will not pop up so quickly. So when you have the antibiotics in the medium, it reduces the contamination, but it does not delete it all. So if you don't have contamination, if you don't have antibiotics, an early stage contamination with bacteria will soon be visible. So the chance or the risk to spreading the contamination throughout the incubator or throughout the lab is reduced this way.
Now our next question. Would you recommend a UV light for sterilizing the cell culture room? For example, leaving the UV on during the night for about 2 hours.
The thing with UV light is that the UV bulb has a limited lifespan, so the effect of the UV decreases with time. So that is one thing that you have to keep in mind. The other thing is that the effect of UV is limited in space, so only items or equipment that is in 50 centimeters direction from the UV bulb will be affected by the UV light itself. So decontaminating the whole room by UV is probably not as effective as it should be or as you think it is.
Now, Dr. Wagner, this next question is a two-part question. What are the sources of contamination, and is it surroundings or the personnel?
Well, it is both. I would say that the personnel itself is maybe even the higher risk of contamination, because the surroundings, like the equipment, when it's regularly cleaned and thoroughly cleaned, is not such a big source, but the personnel itself, goes out and comes back again a couple of times a day. So you have to take care of constant aseptic techniques and preventive hygiene every time you enter the lab. And as people not always do that, or sometimes when you are in a hurry, you are not so conscious about these measures that you should take when you enter the lab. So I would say the personnel is even the bigger risk.
Now, what disinfectant can be used for fungi?
As I said, alcoholic solutions are not the best. So I cannot recommend a special disinfectant here, because there are different ones available. I would just take care that you use something that has a fungicidal effect. And if you're not sure what to use, I would just go in contact with a disinfectant supplier, and they are the experts and can recommend you something.
Now, how often should I perform a mycoplasma test?
That depends on how big your lab is, how many people are working there, how skilled they are, how good they are trained, how good they are in aseptic techniques. So I would recommend to start with a higher testing frequency. For instance, if it's a big lab with a lot of cell culture beginners, I would even start with maybe every 2 weeks or every 4 weeks. And when you have ongoing problems with mycoplasma infection, I would stick to that high testing frequency. But I cannot recommend one frequency that goes for everyone or that works for everyone, because it's really dependent on how trained the people in your lab are and how they stick to their aseptic techniques.
Now, Dr. Wagner, is there any antibiotic against mycoplasma?
Most antibiotics don't work with mycoplasma because they are targeting the cell wall of bacteria, and since mycoplasma don't have a cell wall, they are not effective. There are some reagents that that claim to be effective against mycoplasma. But again, I would recommend to ask the supplier of these reagents for support here.
Now, is it possible to eliminate fungi contamination from a culture, or is it better to discard the culture?
I would always recommend to discard the culture because it is possible to eliminate fungi by using by using reagents that kill the fungi, but these reagents will also always affect your cells. That's the same for bacteria as well. You can treat them with antibiotics and fungi with antimycotics, and you can use a high dose of different of these antibiotics and antimycotics to fight or to kill the contamination. But this will always also be harmful to the cells. So I would only do that as a last option when you have any cells that are too valuable to discard and which you maybe cannot reinitiate from frozen stock so easily.
Now, I find it really very challenging to chase the source of contamination to manage. Do you have any helpful hints?
I completely agree. It's definitely very challenging to find the source, and a lot of times you won't be able to find that one source of contamination. I would just do as much cleaning and disinfecting as possible. So I would start with frequent contamination sources like the water bath. I would completely clean that, and also the water tray inside the incubator, completely remove the water, clean it. If you have the incubator with automated disinfection routine, I would run that. And then all the items that are frequently used, like the pipettes, I would use new fresh tip boxes, and if you can, also fresh reagents, fresh media bottles. And there it is a good idea if you aliquot your medium. Of course, this always depends on the throughput that you have, but it can be a good idea to aliquot the medium. So if you have a contamination and you think that it may be the medium that is contaminated, you can discard just the one aliquot and don't have to discard the complete 5 ml bottle. So that would be one option as well. But yeah, as I said, it is definitely very challenging to find that one source of contamination.
Now, Dr. Wagner, what do you recommend to do when you see a contamination?
I would always recommend to discard the contaminated culture immediately, because the risk of spreading the contamination throughout the lab is high. For instance, when you don't have culture vessel with a tight lid. So when you work a lot with dishes and with plates, these have loose lids. They have to, because you have to allow the gas exchange, and therefore you also have a higher risk than with a tea flask with a tight screw cap. So yeah.
Now, which is the higher risk of contamination? Is it during the collection or during processing?
That depends on the aseptic techniques, I would say. If you are good in aseptic handling, you have less risk during the processing. The collection, when you isolate cells freshly from tissue, always there's the risk of contamination. There it maybe is dependent on where the tissue comes from, how you isolate, and but as I said, you have no chance to sterilize the tissue, because then you would also kill the cells. So if you have good aseptic techniques, I would say the processing of the cells is less risky, but it's different to give a definite answer here.
Now, how often do you recommend to clean under the grill of the BSC?
Again, like all recommendations for cleaning schedules, it depends on how many people are working at the BSC, how many people share that one BSC, and how many, maybe not so experienced people you have in the lab. I would say every 4 months, and then you see what you find under there. If you have still ongoing problems with contamination, it's always a good idea to have a more strict cleaning schedule and higher frequencies of regular cleaning.
What is the ideal number of people who can enter a cell culture lab?
There is no ideal number. I cannot say with 2 people, 2 people would be ideal, 5 people is not so good. The ideal number would be 1, because then you are always sure that you are the person who has to take care of your aseptic techniques and no one else. But there is no ideal number. It's the less the better, but usually you don't have much choice in deciding how many people can enter the lab. One thing that I always recommend is that only the people that have to work there should be allowed to enter it. So, for instance, when you want to discuss any experimental results with your colleague, you should not do that in the cell culture lab, but rather use another room to discuss there and really only do the handling of cells inside the lab.
Now our next question. How can we avoid contamination by reagents?
As I tried to explain during the presentation, you can do some kind of intake quality control. So when you get the reagents, for instance, like FBS, you can culture this with medium for a couple of days without antibiotics, and if there is a contamination in that reagent, it will pop up within a few days. And so then you know that it is contaminated.
What is the best and easiest way to discard the contaminated bacterial culture?
Discard it and immediately autoclave it. Once it is autoclaved, it is not a risk anymore.
Now, looking at our next question, how often should we change the filters in the incubators and clean the fans in the incubators?
If you have an incubator with a fan inside, you have an incubator with a higher risk of contamination than without a fan, because not only that it forces the airflow and therefore is able to spread the contamination throughout the incubator, the fan itself also should be cleaned. I would take a look into the manufacturer's, in the operating manual to look for any recommendations there. I would recommend to do it every 6 to 8 weeks, maybe even at a higher frequency when you have ongoing problems with contamination. The filter that was mentioned in that question is there's probably that is probably the HEPA filter that is inside some incubators. So this should be exchanged twice a year, because you have to keep in mind that it does not kill the bacteria or the contamination. It only traps them. So I think that these HEPA filters, I'm not sure about any manufacturers, but most of them don't withstand the high temperature disinfection routine. So you have to take the filter itself out before you do the high temperature disinfection. So you have to take care that you clean the housing of that filter before you put it in after you have run the disinfection cycle.
Ladies and gentlemen, we are getting so many good questions today. We do only have time for a few more, but just as a reminder, the questions that were not answered today will be answered by the speaker via the contact information that you provided at the time of the registration. So let's continue. What about autoclaving tip boxes and other items? Isn't it a good way to reduce contamination?
That depends. If you purchase tip boxes that come sterile, there you can, for instance, from Eppendorf, there you can rely that you have sterile equipment. Of course, when you use non-sterile tips or tip boxes, you should autoclave them to be sure that you have safe equipment.
Now our next question, which cells are more frequently contaminated with mycoplasmas, primary cells or continuous cell lines?
It is continuous cell lines, simply because the fact that continuous cell lines are cultured for a longer time. So primary cells have, as you know, a limited lifespan. So the chance to get in a mycoplasma contamination is lower, simply because of the reason that you don't have them for ages in the lab, and you don't, so unlike cell lines which you are cultivating over years or decades. So primary cells have the lower risk.
What if the cell culture which is contaminated is very valuable and expensive? Is there any other option to decontaminate them?
Yes, as I already mentioned, there is, you can decontaminate them by treating them, or you can try to cure them by treating them with high doses of different antibiotics. But if you do so, you have to keep in mind that these high-dose antibiotics might also affect the cells themselves. So I would always recommend, if you have done this treatment over a certain period of time, then to proceed the cultivation without antibiotics to see if the contamination is really gone. And if so, I would give the cells a few passages to be sure that the effect on the cells is not so drastic. So I wouldn't use the cells directly after the treatment for any important experiment. So give them some time to recover from that treatment.
Now, should two people be allowed to use a BSC simultaneously?
It is always better if there's only one person working at the hood, because the more people are moving their arms there, the more turbulences you have in the airflow, and the higher the risk is that you get germs into the BSC. But of course, sometimes it is not. You cannot avoid that you have two people sitting there at once, because the day only has 24 hours and people need to do their work. So no matter how you do it, I would always take care that you only have the items in the hood that are needed there. So don't overcrowd the workspace, no matter if there's only one person or two sitting there working. And they should try to yes, they should try to avoid any hectic movements and too much of going up and sitting back again, and try to prepare the workspace as good as possible and put everything in there that is needed so that you don't have to stand up again and again to get everything there.
And it looks like we have time for one more question. What should we clean the incubators with to ensure no residue?
We recommend for our incubators to use 70% ethanol or isopropanol, because, as I said, these disinfectants are the only ones that evaporate completely residue-free. So for other contaminants, for other disinfectants, you always have the risk that you have any residue that will, stick to the material and does not evaporate as alcoholic solutions would do.
Thank you again, Dr. Wagner. Do you have any final comments for our audience?
I want to thank you for your attention, and also thank you for all the very interesting questions that were coming in, and I will take care that I answer all the remaining questions later offline. So watch out for an email that you will receive within the next couple of days.
Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank Dr. Jessica Wagner for her time today and her important research. We would also like to thank LabRoots and our sponsor, Eppendorf, for underwriting today's educational webcast. This webcast can be viewed on demand through April of 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.